Hi, everyone. Good afternoon. If you're on the East Coast, potentially good morning or good evening if you're elsewhere. So today we have a wonderful presentation by Professor Carlos Bustamante at Stanford University. And we look forward to a great presentation. For the past 18 years, Dr. Bustamante has led a multidisciplinary team working on problems at the interface of computational and biological sciences. Much of their research is focused on technology and its application in medicine and agriculture and evolutionary biology. His first academic appointment was at Cornell University's College of Agriculture and Life Sciences. There, much of his work focused on population genetics and agricultural genomics, motivated by a desire to improve the foods we eat and the lives of the animals upon which we depend. He moved to Stanford in 2010 to focus on enabling clinical and medical genomics on a global scale. He is focused on reducing health disparities in genomics by one, calling attention to the problem raised by greater than 95% of participants in large-scale studies being of European descent, and two, broadening representation of understudied groups in large NIH-funded consortia, particularly minority groups from the US, the Americas, and Africa. He was the inaugural chair of the Department of Biomedical Data Science and a founding director of the Center for Computational, Evolutionary, and Human Genomics. His work has empowered decision makers to utilize genomics and data science in the service of improving human health and well-being. And in the next phase of his career, he'll focus on opportunities for bringing these technologies to consumers, patients, and patients directly, where this work can have the greatest impact. And with that, I'm going to hand off the baton to you, Dr. Bustamante. Thank you for joining us today.
Thank you so much, Harry. It's a real pleasure and privilege to be with you guys. Can everyone hear me okay? Does it sound all right? Yep. All right, perfect.
Sounds good.
Good. So, I want to take the opportunity today and really tell you a little bit about the intersection of two lives. My life as an investigator and scientist at Stanford and building consortia to study the genetics of first the human diasporas and then the complex diseases that we and our relatives suffer from. And then the second is as a real active participant in starting companies and investing in companies. And part of what I really want to talk to you about is why what has been, I think, really well described as a three-player game is so critical to success in diversifying the basis of what we think we know about human physiology and variation. And so there's a very strong business argument that I need to make, and partly because eventually the only way you're going to get resources is by making a strong business argument. You know, political arguments are good, but they're nowhere near as good as business arguments. And so I wanted to call your attention to a just extraordinarily smart book by a Keynesian economist called Bill Janeway. So Bill was a practicing venture capitalist and a what you might call demand side economist. So he was in the British tradition of John Maynard Keynes and the importance that the state plays in really pushing the economy forward. And he described it as a balance between the state investing in early innovation and we think of that as Bohr's quadrant, right? Investing in basic science, the building of the knowledge base. Then there's an opportunity for speculation, financial speculation that would bring in and bet on technology. We often think about that as the start of markets and bringing competition to bear once something's left the Bohr world and entered into Pasteur's quadrant of really being useful in society, having improved use, having real market for products and services, and then ultimately you're playing an Edison quadrant, right? Namely, innovating and staying a step ahead of the competition. At which point you're really just scaling out operations. And I want to think a lot about healthcare in that context, because healthcare is a business like any other business. In fact, it's the biggest business in the country. The United States has always been about business. And so if you really want to understand healthcare, you've got to sort of be focused on that. And there's a very strong argument in that framing to make about diversifying the basis of clinical trials. I'm also a conflicted investor. And so I want to both proudly back and mention the companies that I've put my money behind as well as my sweat equity behind. I'm a firm believer, I'm an immigrant to this country, I'm a firm believer in the ability for new firms to enter and outcompete existing decrepit industries and other firms that can be overtaken. And so I like to bet on the underdog, and sometimes they take off. So we were the first advisors to a company called Invitae that's now one of the largest producers and providers of next generation sequencing tests. Also Personalis, which some of you may know, was spun out of Stanford. I was Ancestry DNA's first advisor. I've also played some in medical cannabis, for example. Some of you may know Columbia Care. It's one of the biggest producers. I helped build a lot of their medical advisory group and strategy. And then I've played a ton in animals. I think animals are an incredibly important place to learn about genetics and markets, and largely because they're deregulated. I think they often deliver much better care. So I say my dogs get much better care at VCA than my kids get at Stanford Med. And it's a large part because veterinarians understand they have both a patient and a customer. The patient is the animal, and the customer is the pet parent. And many of the things that you'd like to see happen in human medicine are actually happening already in animal medicine, including, for example, liquid monitoring at hundreds of dollars for pet cancers, the ability to target therapies to pet cancers, the ability to improve the lives of horses and dogs in eradicating genetic diseases. I mean, there's just really a wonderful world that's coming up front there. And I think a lot of the technology will spill over. So why is this at all relevant? Well, the problem I've worked on my entire career is really a problem of how do we enable precision health at scale? And that's not an easy problem, especially precision health at scale for all, and I want to frame it in my wheelhouse. So, I am first and foremost a population geneticist. And so I want to start by saying, I really think human genetic variation plays an important role in health and disease. I think studying human genetic variation is an important thing to do. It has been an extraordinarily rewarding thing to do for me as an investigator. And I think it has helped improve the lives of the populations that we studied by, bringing attention to the first, beauty and diversity of human phenotypic variation. So for example, we've mapped the gene for blonde hair in Melanesians and shown that it's a different gene than blonde hair in Europeans. We've built massive networks to get at access to samples from all over the world to improve the basis of what we think we know about the frequency of disease alleles. And then we spent a lot of time working in, real historical problems, including the transatlantic slave trade and reclaiming parts of human history that have been lost. And putting that back in the hands of those that are the descendants of those folks. I'll start by saying that to me this came from a long fascination with human evolution and human migration, and again the just beauty of human phenotypic variation. When I was in graduate school, the view, in fact, early when I started as a professor, the view was one summarized here by Cavalli, Sforza, and Feldman of the, out of Africa model of human origins and subsequent bottlenecks and the great human diasporas. That has now led us to just create what really was the most incredible collection of diverse samples from throughout the world, including things we had collected, things the Human Genome Diversity Project had collected, things that others had collected. We led a lot of the effort in 1,000 Genomes to diversify collections and do it in an ethical way and collect samples with consents that could be used to create cell lines, and cell lines that could be used not just by academics, but by companies as well. So, we've really put resources behind creating community resources, which ultimately is one of the most important things that we can do. So I would say, the current view is one of just richness in the history of our species, right? I mean, we are a wonderfully complex and successful small-bodied primate species that over the last couple 100,000 years has really just overtaken the earth. And it is a mesmerizing feat of human adaptation and human ingenuity. And it has led, of course, to signals in our genome that both tell us about that history and tell us about why parts of the genome have evolved in certain ways, largely driven by the foods we eat, and the response to pathogens. And this is a story that in many ways belongs to all of us. We've been telling this story forever, right? Where do we come from and who are we? And genetics is just the latest layer in that tale. And so this is a project that literally began more than a decade ago, and many of these people are now extraordinarily successful in their own right. So John Novembre is now a full professor at the University of Chicago. He was a postdoc when we started this. Matt Nelson's now running a huge, genome center out of GSK. Matt Nelson, was my partner, but a lot of this were folks in my lab. Adam Otten here is now the vice president of genetics at 23andMe. Adam Boyko is a professor at Cornell, and so on and so forth. So, it really has been a lifelong mission for many of us. And the first data I want to show you is like where we started. This was AFI 500K data before anybody knew any better. And what we did was really bring to the table what were standard tools in informatics, which is part of the story I want to tell you, right? You have an extraordinary tool set, and that tool set is tactical and can be deployed strategically if you know what kind of questions you want to ask. So in our case, we were looking at this really weird 500,000 dimensional by 4,000 dimensional matrix that had 0, 1s, and 2s, which were the genotype data. And we didn't know what to do with it because all the beautiful theoretical population genetics that I had built as a graduate student and postdoc that required sequencing and random sampling was totally useless, right? This was heavily ascertained data. You could not build proper population genetics models. We couldn't do, Lee and Steven sampling. We couldn't do the full-blown coalescent. So we had to go old school and just pull stuff out of the hat. And the first thing we pulled was principal components. And oh my gosh, if it didn't just crack open a really interesting part of the puzzle, namely, the genotype matrix really told us about these out of Africa and subsequent migration. So much as we've known for a long time, the vast majority of human genetic variations within Africa. So it makes sense that the first principal component would be an Africa out of Africa principal component based on our common human origins in Africa. The second principal component separates Europeans and East Asians, which again makes a tremendous amount of sense given what we've known historically from the peopling of the Eurasian continent. And you'll see, for example, that African Americans on this plot lie in between the Yoruban centroid and the centroid of samples from Europe, namely because they have an admixed ancestry. They have, on average, 20% European and 80% West African ancestry, and literally the distance in PCA space tells you that, right? So this was really powerful. This was not kind of old school Cavalli Sforza, let's take populations and PCA the frequencies. These are individuals that you can now rotate in genetic space and find out who their closest relatives are. Furthermore, we could continue down the fourth principal component. The third principal component separated East Asians from South Asians. And then the fourth principal component separated the East Asians from what must have been an unsampled Native American population because we're seeing this principal component really only impacting individuals of Mexican ancestry. And we were the first, this is like literally the first genotype data on Hispanics. It was the first genotype data on South Asians. This was like just as HapMap is coming online, giving us a far bigger richness given the number of samples that we had here. So where this story went, as many of you know, is to focus on the small blob of European diversity. And you might say, well, that's a reasonable place to start. However, as John showed in his now iconic 2008 paper, even Europe has a tremendous amount of variation. In fact, that variation lies along geographic lines that we've long understood. So the first principal component is a north-south gradient from the US, from the UK down to the boot of Italy, and then east-west, it goes through the Iberian Peninsula out into the Caucasus. So in many ways, this was an incredibly interesting result to get, but it also illustrated why this notion of trying to put boxes around individuals is cut and soup, right? Even a country like Switzerland has diversity and variation where the French-speaking Swiss are genetically closer to the French, the Italian-speaking Swiss are closer to the Italians, and the German-speaking Swiss to the Germans. So this is really about parentage, and it's about really the local structure of who is related to whom. We've then, I think, evolved this into a view of what one might call clan genomics, and the idea that alleles are going to percolate in the human gene pool at different, entry points in time. So there are ancient high frequency segregating alleles, right? This is the bread and butter of heterozygosity, things above 5% frequency, and we've known a lot of that variation. But of course, the vast majority of variation is extraordinarily rare. It does not contribute to heterozygosity, and it is, potentially the source of what may be missing heritability in some of the complex disease. And so if you really want to create a fair system for all, a precision health for all, you cannot ignore the corners of the diversity space. And that's really the most important message that I have to share, right? Having looked at, reams and reams of genetic data, studying only a small corner of genetic diversity space is the wrong thing to do. And I've been calling attention to it for a decade, as well as trying to figure out what can you actually do about it. It's one thing to say, well, this is wrong. And another thing to say, well, this is what you should do about it, And so what we've tried to do is stay really close to the core of the science, which is the biomedical data science here, and let that lead our thinking, because it's ultimately through the distillation of data and the creation of platforms at scale you can actually move health, right? We really want to move health from a artisanal delivery process to an industrial process. That is what we want to do, right? We do want to mechanize the delivery of healthcare and specialize it based on indicators. And to me, this is literally a supply chain problem, right? This is how do we optimize across a complicated space. And as a methods developer, I'd like to tell you that it should all be methods and methods and methods and methods. Unfortunately, methods are only, again, tactical. They're only going to help us solve optimization problems. They don't tell us what optimization problem to go after, right? And in many ways, what I want to tell you is that it's really all about creating new objective functions in the US healthcare system. And I'll start with what I think is the right way to do things. So this is the work of colleagues in Vanderbilt. Some of you may know the work of Josh Denny and Lisa Bastaracci, who have demonstrated the ability to mine the electronic health record data and identify patients at risk. In their case, they were looking for Mendelian disease patients. I encourage you to read this paper. And the logic here is that I can stream electronic health record codes once I know somebody's genotype and use that to figure out whether the EHR stream contains enough information to separate that individual from a background set. And so in the case of, say, for example, individuals who are Delta 508 homozygotes, right? You might look at their EHR and say, what kind of resources do they utilize? Or somebody who's a heterozygote or a compound het, and begin to build a signature, and then go back through untrained data and try to identify undiagnosed patients. And the key is there's plenty of signature in the healthcare system to do this. So they did this for cystic fibrosis, they did this for Marfan syndromes, achondroplasia, and so on. The interesting exception that proves the rule is PKU, because there are plenty of people who are genetically at risk of PKU, but who don't suffer from PKU because you can put them on a specialized diet, right? So this logic of being able to go through and use the electronic health record system itself to identify patients at risk and put those patients on specialized therapy is at the core of what we want to do in precision health. However, in our setting, a disproportionate level of chronic disease burden is found in minority populations. So if we want to go out and really make a dent, we're both understudying the populations we want to work in, and at the same time, not necessarily addressing what is a growing issue, which is the changing face of both the country and the world. So we began, as I said, in 2009. At that time, there were something like 1.7 million genotype samples. 96% of them were of European ancestry across 400 or so odd studies. Several years ago, Alice Popejoy revisited these numbers. There were something like 35 million samples in the GWAS catalogs. 80% of those were of European ancestry. There was a massive increase in samples of Asian ancestry, largely due to the entry of China into genomics, but nowhere near the concomitant increase in populations of African American or Hispanic descent. So that to us is a big issue, and it's a big issue, as I said, because of the changing face of the country, right? So if we look at where the country's headed, we are headed to a far more diverse, far more admixed world, and it's just unconscionable that the basis of our research studies are outdated, particularly because it takes years to translate research into clinical outcomes. So it's this just like runaway process that unless you proactively go out and oversample, it's just naturally going to get worse. Technology is just naturally going to make health disparities worse if you don't address this at the core. So I want to walk you through what can you do about it, right? So it's one thing to call attention to it. What can you do about it? I'll quickly run you through the largest study to date that's been federally funded, which is our study called the PAGE Project, where we literally emptied all the freezers out of the NIH and took it to town. So this was published now almost 2 years ago. This is led by my former postdoc, Genevieve Vacek, and was published in Nature. And it was literally the largest collection of African-American and Hispanics genotyped for cardiometabolic traits to date. And the design of this project is really quite something to be proud of. It really is Jen's work and the rest of the PAGE project. They from soup to nuts conceived of this project, including the building of new resources, new cohorts. We began with, again, just the stark reality. UK Biobank's 94% Caucasian. Same is true of Emerge, same is true of Giant. I mean, it's just the reality of the studies that have been done to date. So at PAGE, we went out and worked with USC, which had the multi-ethnic cohort, UNC, the Mount Sinai cohort, and built, a 50,000 person cohort that was predominantly Hispanic, African-American. We had the first Native Hawaiian data, and we built a special chip for it. So it wasn't just enough to take a chip off the shelf. We literally built the MEGA array, which stands for multi-ethnic genotyping array, commercialized that through Illumina. That became the core of the global screening array. And with MEGA, we were able to go after these 26 traits simultaneously. I'm not going to go through and give you each of the GWAS plots because, nobody wants to do that. But let me just give you kind of the highlights of what we're, seeing here, namely that it makes a huge difference to be able to look at this across different populations. So here, for example, are the anthropometric trait where we found, new hits. We've got, inflammation, we've got glycemic index, we've got lipids. And perhaps, one of the things that you might want to know about is, do we find things that we had no ideas? Are there new genes? Absolutely. So, CREB3L2, we find due to a large, frequent variant in African Americans that has a significant odds ratio, of course, at the sample size that we've sampled. And you would not have found this in Europeans because it's at 0.5%. Right. I mean, that's why you didn't know about it. Right. And so just adding African Americans immediately lists, massively lifts the list of genes to look at. And within that gene, of course, you're going to have the most power to now go back and actually re-genotype this in European population. So it's actually like, the scientifically the right thing to do, if not just the ethically the right thing to do. And when you put this together, I mean, the replication power is extraordinary. The fine mapping power is extraordinary. I mean, it's the reason the paper got published in Nature, right, with 50,000 samples. And again, what the logic here is that by increasing the number of recombinations in the history of the set of samples, you can fine map better by bringing diverse cohorts together. Their ancestors recombined back, 150,000 years ago, and that just really narrows down the region of association. And to me, why this is important is, because we are phenotypically and genetically diverse species. We cannot just, pretend that everybody is identical. It's just not true. And we wouldn't want that to be true. We relish human phenotypic variation. It has a genetic basis. We have to see this as an incredibly important asset of our common endowment as humans. And so the ability to study that is both a gift and an opportunity. And, we've shied away from it out of, I think, really just bad policy. And so we've been striving to change the policy and move us in the right direction. Now, this is a project that, again, took millions of dollars and a huge government enterprise. And we can't do it again. We literally can't do it again because it was years of samples that were already collected, right? So we've shaken the freezer. There are no more samples, no more samples. So the All of Us cohort is starting again. They're trying to collect several 100,000 people, follow them longitudinally. But, it's going to be years before we can reach the kind of capacity that you saw in the UK, right? If you turn around and you look at how the United Kingdom handled the COVID epidemic, it should give us pause. It should give us pause as a country. Here is a far smaller country that was able to turn around and use its healthcare system for research and illuminate to the world how you could solve, a new challenge to the human condition. We couldn't do the recovery trial in this country. We don't have the infrastructure to do the recovery trial in this country, but we could build it. And part of the reason is, biomedical data science is not an objective function, right? Like when we think about data and the need to munge on data, it's literally a question of what do you want to do with it? And if there's only so many hours in the day, I spend it looking at this figure, which is a figure I made for myself several years ago. And it's nothing more than a little cheat sheet about where I think the incentives are and where the world lives. So when the dean asks me to start a department of biomedical data science, he's up in this top corner. He cares about a business. He runs a big $2 billion a year business. So he cares about his electronic health record, omics, imaging, pathology, outcomes. How do I give better outcomes, improve the bottom line? Of course, I mean, you'd be an idiot if you're running a business and not trying to do business analytics. And that's the software we've built, right? We've built really good billing software to optimize the business of healthcare. Why? Because that's what's incentivized, right? Like let's be adults about that. Right. And if we think about what is the interaction between the software we've built and capital, it's healthcare economics. And it's literally how much should we pay for this procedure or that procedure or this thing or that thing. It's all reactive, acute, sick care. It's not healthcare. It's not preventative. It literally cannot be incentivized to do that because the structure is in a totally different domain. So if you really want to think about healthcare and you want to move people, we all know it's got to be about patient reported outcomes and about the data people have and carry for themselves. And this is not anything new. It's just that, well, we've taken long enough, but I think the future's here, right? And COVID has accelerated the future. So there's no longer a really good excuse for why your EHR data isn't integrated with your apps, because that's easy to do. Right? And that's coming. And there's no reason for you to be doing clinical trials and not integrating digital streams and doing real world evidence and doing omics, because that stuff's easy to do and it's the future. And it's the only way that we're actually going to move the needle. I'm a firm believer that the kinds of things that 23andMe and GSK laid out several years ago were exactly the right things to do. They just IPO'd, as you know, several weeks ago, and the logic is super tight. You can absolutely use genetics to improve the bets on drug prediction, and it's not that difficult. It's an incredibly inefficient process. It takes $2 billion in 10 years to bring a drug to market. If you can use genetics to improve it a little bit and save hundreds of millions of dollars, people will listen to you. And the best example I've got is PCSK9 inhibitors, where we went from bench to bedside in 6-7 years. So this is, from 2015, the New York Times hailing the new breakthroughs in cholesterol lowering drugs. These come from our ability to understand people with extreme genetics. It begins like any genetic study with a quantitative trait. In our case, it's bad cholesterol. And you have individuals with high bad cholesterol and individuals with low bad cholesterol. Well, if you get really, really high bad cholesterol for genetic reasons, you can even get a condition called familial hypercholesterolemia, which is due to, one of 3 different genes. And if you are homozygous for, 2 alleles, you can have a 20-fold higher risk of dying from an early heart attack or stroke. And this is not rare. This is 1 in 200 people. So the study of familial hypercholesterolemia has been at the core of a lot of what we've learned that has actually allowed us to create better drugs. And one of those are these PCSK9 inhibitors. And the logic here is that individuals with, familial hypercholesterolemia are from birth accumulating this risk. And so it turns out that if you have one allele by the time you are, in middle age, you've got the lipid profile of somebody in their 60s. And if you had happened to be one of the homozygotes, then you'd have, the lipid profile of somebody in their 60s by the time you're starting at Columbia. So that's a big deal. And this is a disease that responds very well to therapy. However, 90% of people are undiagnosed. So the reason I'm telling you this story is that it was the study of the genetics of cholesterol that allowed us to identify people with a superhuman mutation. These are people who carry a PCSK9 loss of function mutation. So here's most of us in this room, right? Most of us are on the Zoom call, right? We don't carry a PCSK9 knockout mutation. Our LDLs are somewhere around 125. For those of you that were lucky enough to be born with this, your LDL would be between 50 and 100, right? You could eat as many hamburgers and hot dogs as you want and have really low LDL. More importantly, you've been dosed from birth with a life-saving drug, 80% reduction in the risk of cardiovascular heart disease in individuals with a PCSK9 mutation, irrespective of ethnic background. So this is huge. This is, if you're looking at this from the drug development point of view, the genes that predict bad cholesterol predict whether or not you're going to have a heart attack. Let's go. And in fact, that would have been a really great bet. All 5 drugs that went to market trying to lower bad cholesterol made it out of clinical trials, were safe, effective, and approved. 5 different drugs that were trying to raise your good cholesterol failed in clinical trials. They actually raised the good cholesterol. That wasn't the problem. It wasn't the biomarker. It's just that the biomarker was not predictive at all of the clinical endpoints. So if you're sitting on this biopharma pipeline, let's say you're GSK, you want that genetic data to place the right bets. It'll help you improve the pipeline. Fascinating. What's the problem? Why can't we just turn the crank on this? Well, when they first went to market, this drug cost $12,000 a year. A third of Americans have high cholesterol. You're not going to put a third of Americans on a drug that costs $12,000 a year. Like, we literally don't spend that much on all drugs, much less on a new drug to lower cholesterol when you don't necessarily know that the people won't respond to a super cheap drug like statins, right? So it shouldn't surprise you that while the FH Foundation wants everybody to be screened early and get on therapy early, it's a sad state of affairs in many ways, right? Like, the average age at which people are diagnosed is close to 50. And by that time, they've already been on lipid lowering drugs. They might have already had a heart attack or a stroke, right? So we're way behind where we should be. And a huge part of this is really kind of tilting the incentives. So the end result is that in this country, the vast majority of people who are prescribed this drug never get it. Right? So we've developed this magical drug. 95% of the time it gets denied the first time. Eventually it gets denied 62% of the time. So the vast majority of people who are prescribed this magical drug don't get it, even if they're a high risk cardiovascular disease patient or FH patient, right? Just because the system needs proof that it's worth paying for, right? And I would argue that this is where the data science can really make a huge effort. And huge difference. So this is the work of Josh Knowles and Nigam Shah, 2 of my colleagues at Stanford, who've taken this problem and said, okay, we should build a tool that allows us to, sift through the reams of data produced by the healthcare system and identify folks at risk. Let's start with Stanford. So they built a classifier at Stanford that would allow them to look through clinical notes, conditions, prescriptions, laboratory measurements, and use a random forest classifier to identify presumptive FH patients. They then cross-validated this at Geisinger, but more importantly, they were able to scale out the whole algo by working with the FH Foundation, getting a bunch of de-anonymized data. They could literally build a map of the country and where people are at risk. And the last bit is they created a website where if you're a physician, you can log on, find out from the FH Foundation who your presumptive FH patients are, because you have the secret decoder ring, you're a covered entity, you can kind of uncrack that whole thing. The last piece of this whole story is that I think this is going to accelerate and be out of the university before we know it, if it isn't already. So 2-3 years ago, a company called Color was already doing FH testing and doing FH testing faster, better, cheaper than Stanford. And I would argue this is going to fall into what is a very classic understanding of innovation. And it's just what you expect when you begin to industrialize medicine and biology, namely that much like, the classic example of cell phones and landlines, right? Where you have two ends of the market. You have initially a consumer end of the market, and you have a high end of the market. The consumer improves faster, better, cheaper, and eventually disrupts, right? So, 10 years ago, it would have been unheard of to not to have a business and not have a landline. Today, it's totally the norm. And in fact, the shift in landlines versus cell phones, this is, the crossover in the United States didn't happen until about 2015, 2016, where you only have, landline only in US homes. So it takes a while, but the disruption is there. So my thinking is Helix, Color, these will absolutely come and disrupt and should disrupt the tertiary care business, which is just expensive, out of date, should be disrupted business. And again, this is a cliche now that this is all due to the data warlords jumping in. I would argue that, the biggest companies in the world are data companies simply because of the money they have, right? When I was in college, the biggest companies in the world literally were the oil companies. They had the biggest market cap, they had the biggest cash on hand. Today they're they're the data companies. And so of course they're going to go into healthcare. It's the biggest industry in the country. And so if you're sitting on, and again, this is out of date, if you're sitting on $250 billion in cash in your Apple, of course you're going to gun for the industry. If you're Microsoft, of course you're going to gun for the industry. If you're Alphabet, of course you're going to spin up Verily. And in many ways, this is old news, right? We already know we can get so much from our wearables that it makes no sense to do anything but try to follow our patients using these devices. And so in that sense, the ship has left the port. The academic centers are no longer really in control of these data streams. I think we'll be in control of potentially the algos that come in and make claims. Where does this go next? I think it goes from the genetic sensors to all kinds of additional sensors, right? You could think about not just your genetic data stream, but your LDL data stream, your oxygen and performance data stream, your, quarterly blood test. And this is the future of how, we should be tracking folks. And this is just one company called Inside Tracker, but there are several others. And eventually, I think this also goes into some kind of auditable system where you eventually can put a value on some kind of, quantum. And that quantum might be some piece of information that links either genotype to phenotype, or it links an incentive to an action. But this is, of course, all coming, right? If you have Dogecoin, why wouldn't you have HealthCoin? Of course, you're going to have HealthCoin. There are already a bunch of health coins. It's really a question of how do you begin to put this into a system where the value is in tangible ways, right? And I would say that the logic for a blockchain would be to keep the giant permission file for how people's data can be used. Like that already exists in some sense, in that lots of your data gets sold and resold without you knowing about it. So if folks could know about it and sign up and sign in, then you could enable really interesting contracts that allow lots more data to flow. So we've worked on things like this. Luna DNA is one such effort, but there are others. I would also argue that this is going to be relevant when you think about patient-reported outcomes. So if you don't like PCSK9 at $12,000, then you might not like Spark Therapeutics and Luxturna. So I think Spark has already been bought, but it was a company spun out of CHOP that had a $750,000 per eye therapy that cured blindness. Luckily, it came back with a money-back guarantee. So the way Luxturna is set up is that you pay, but then can claw back if the patient has not met outcomes. And so I think this is one area where you're going to be really careful and make sure that you enroll the right patients, you've genotyped them, you've done the right thing. And because of the opportunity to go into rare diseases in lots of ethnic groups, I think this is going to be a surprising place where you're going to begin to diversify a lot of this work. And that's not even the most expensive. If you don't like if you don't like Spark Therapeutic and Luxturna, Zolgensma is like $2 million a shot. So it's just going to get more expensive. And maybe I'll end here, which is that there's a lot of really interesting fintech that's getting developed around this, including the opportunity to create, almost mortgages for some of these therapies. Maybe you think that's interesting. Maybe you think that's horrifying. But it is part of the calculus that will need to get done. And my sense is that there's no logic to keeping the data locked. It only makes sense if you can use it to break knowledge asymmetries. So with that, maybe I'll stop here and say that healthcare delivery costs worldwide are the biggest challenge to economic growth and human well-being. Being an innovator doesn't solve the problem, and to me, the supply chain problem here is pretty evident, and one where you're going to need to do a massive realignment with investment to sop up what are the consumer lack of demand in genetic testing. So I think it'll basically come to 23andMe Ancestry have already tapped out their customer base. Everybody making over $150,000 a year has had a chance to take a 23andMe test or Ancestry test. But there's a lot of capacity, and there's a lot of interest in figuring out how you turn the U.S. healthcare system into something that can be used far more efficiently for research. I think a national project that begins to layer on genetic information, particularly people who got COVID, for example, would move us in the right direction. Think about a UK biobank project that started with COVID positives. That would oversample the poor, it would oversample minorities, it would oversample people with comorbidities. I literally think it's going to be massive infrastructure projects like that are going to get needed, and you almost go into a war economy where you're hiring companies to do things that the government should be able to do but clearly can't do. And unless you use the combined purchasing power of the government and big pharma's interest in diversifying clinical trials, there just literally won't be the scale necessary to lift this. So. With that, happy to stop, take any questions. Hopefully this was in the ballpark of what people wanted to talk about.
Thank you so much, Dr. Bustamante. This was fabulous. It absolutely, I will say, was in the ballpark of what people, are interested and want to talk about. So we'll open this up to questions now from the audience, and please speak up if I can't, see you raising a hand.
I had a question. Thank you so much, Dr. Bustamante. My name is Pierre Elias. I'm a cardiology fellow currently doing my postdoc in the lab of Dr. Adler-Perot here in DBMI, and I focus on deep learning with cardiac imaging. I was really interested in some of the things you were bringing up. So one paper that we've talked about writing for a while is, can AI be anti-racist? And I think we've seen tons and tons of papers about implicit bias that can come when you're generating models and you're not paying attention. The thing is, as a practicing cardiologist, I can say, our current medical practice is very racist. One area that definitely comes to mind and was kind of our use case is cardiac amyloidosis, which is this blameless disease that you develop from a very early age because of a misfolding protein in your body. And then by your 6th or 7th decade of life, you've got heart failure and you either need a heart transplant or a $200,000 medication. With our population here in Washington Heights, Matt Maurer's clinic, who's kind of the godfather of cardiac amyloidosis, should be filled with people who are Caribbean, Hispanic, and African-American, but it's a bunch of white people from Jersey. And the reason is because you got to see 3 or 4 doctors before someone kind of puts the gears together and says, oh, this is cardiac amyloid. You need to go see a specialist, pyrophosphate scan. And so we want to show that deep learning models, if you look at the people who are getting cheap.
So maybe I'll interrupt and say, it's a little bit like long haul, right? You have to have a certain amount of money to get classified as long haul. Although Miss C hits all the minority kids, right? So like, how does that work out? And it's like, you have to have so much money to actually go out and be able, same thing with, recurrent Lyme disease or PANS panda or chronic fatigue, right? It's not like these don't affect minority, just like those people unfortunately don't get registered at all and probably have horrendous outcomes. You have no idea what happens to those poor people, right?
Yeah.
And one of the things that COVID has taught us is that a very simple lesson: not everybody responds the same way to an infection. like a really important lesson. And maybe that like has given a lens of empathy that you can walk through this, because my logic is, boy, I think we can all agree if you got COVID and got really sick, it's not your fault. Right? Like no matter what, like if you got COVID, it literally, this should have never left. It shouldn't have happened. Right? This is like we've done this global screen for this horrible virus, and it's just a tragic natural disaster, like a hurricane, right? Or a wildfire. So these poor people who've gotten sick, who've gotten recurrently sick, they should be at the front of the line, right? And I think as a consequence, you'll say, oh my gosh, and like, how are we going to manage their diabetes? And oh my God, how are we going to manage all of the, comorbidities that are really just like what ends up happening to you if this is the sort of lifestyle that you end up living, because of, the course of your life. So anyway, I didn't mean to interrupt you, but just to say that I think it's really interesting how different diseases are stratified by income. And I see it on the genotype side. Like you literally go out, 50% of people making over $150,000 have already been genotyped, and it tops out at 60%. So like the ones who haven't been genotyped, like, I don't want to get genotyped. I think it's creepy. I don't want Ann Wojcicki to have my data, right? It's not like I don't have the money to pay for it.
I completely agree. The thing I wanted to ask you is, as we think about developing research in this area, one thing that will always be true is you have an imperfect sample. You're not going to go and get a randomized clinical trial built around this. And so people will always be able to say, oh, well, the reason that, your model is finding more people of minority populations that have this disease than what's going on currently is because they're not the same people, right? The average age of your population by race and ethnicity is older in Caribbean Hispanics than it is in whites and things like that. And I'm sure as you were trying to develop this work to say, look, there's something here that we've been missing, you face similar challenges. And I was wondering just here, how do you construct a perfect study from imperfect data when you're trying to find this, this implicit bias in practice, which can be a difficult message for people to accept?
So just two comments. First, and trivial but important, right? Don't let the perfect be the enemy of the good, right? And done is better than perfect. And so we also have perfectionism, and that can be, that can inhibit us from even starting. At the same time, we also want to be super careful not to fall into the same epistemological traps that other people fall into, right? Like, and so what I've done in my line of research is to focus on problems that are properly powered, and in fact, sometimes overly powered, so that if you do have systematic issues in the data, like they'll scream at you. So when we did, for example, the PCA work on GlaxoSmithKline, initially the first principal component was the day the samples were run, right? Because there were just bad SNPs that you have to go in and clean. So there's just a process to doing data science around data cleaning, around data, not reusing data, not dilute, so if you use best principles, and then you can weave your way through to causal inference, even in imperfect samples. I mean, that's the whole reason to set things up. And then to the extent you can, cross-check things. So recovery trial is great because it is randomized control. So like we should be able to check some things there, and then we should be able to redo something. Okay, other questions? I want to make sure to get to lots of voices here.
Like Vijendra has a question.
Oh, great.
Oh, hi. I'm Vijendra. I'm a third-year doctoral candidate in Nick Tatinetti's lab, and I had a more conceptual question based on getting trials like these or studies like these up and running. And I like that you brought up the NHS in the UK in terms of how they got their study, the recovery study going. How do you go about doing something that large in a country where the healthcare system isn't nationalized, much less the medical record system? And there's always the issue of the actual patients not all being on the same, or not all being in the same position in terms of this is for a larger, better understanding of health and not some conspiracy.
Yeah, so there's a lot there. I would say that maybe I'll take the in two directions. Well, one is that vulnerable populations, really are important, and we want to make sure that their rights are protected, right? And so, one of the areas where genetics, I think, has been rightly criticized, for example, is in treatment of indigenous populations, where there's a real clear knowledge asymmetry between the investigator and the groups that they're interacting with, and so on. That's also true in medical research, right? And we don't always kind of view it that way, but we do have a lot more knowledge than our patients and participants. And so I think the thing that often worries me the most is what I call knowledge inequality, because I agree that it's easier to like give people the resources they need to be able to achieve what they can do, than to have to go in and then also deal with just a ton of negative externalities that make it even harder to do that. So the sociocultural framework is one that is incredibly hard and has to be done right. Where we've done it in the anthropological setting is literally like, I had a rule: I wouldn't go and work in a place unless I had an anthropologist on staff who was a specialist. Like, so we did work in South Africa, we did work in Mexico, we did work, and it just felt like just the right way of doing things. In US minority populations, most of what we've done have come from, again, these large pre-existing cohorts. And when we do recruit, we then try to switch and almost err on the side of going to urban populations, almost college students, right? Like if you look at 1,000 Genomes, you went to college students, you weren't asking them health questionnaires. So there was like, the risk was low. So you try to create spaces where the risk to participants is low, you re-engage with return of results. And I don't think that that's like all that different between anthropological genetics and trying to get, people in urban communities to participate in research. Like, I think a lot of that is going to be shared in common, and and in many ways, rural and indigenous populations are just as, of course, they're just as modern as we are. And I used to say, like, I want to kind of wait until I can pay Masai in Bitcoin for them to sign a smart contract that they understand and want to share their data with me. And I don't think that that's that far off, right? And that's actually the fair thing to do. You almost feel like the decentralized finance movement will get there first and enable you to meet participants and actually engage them in fair contracts. Not sure if that answered your question, but I do think it's where some of this goes and how we learn how to do this.
Great. I think we have time for at least one more question.
If no one else has a question, I'd like to ask one.
Go for it.
Yeah, so I think there's been, sorry, my name's Oliver. I'm a fourth-year PhD student in Noemi El-Haddad's lab. I was wondering, so there's a lot of talk about like participatory research, getting people into studies as subjects and everything, making sure that's a diverse population. But I'm wondering if you've seen any pushes to not only have a diverse like research subject group, but also diverse like people who are doing the research. So Dr. Kielo Fox has a really great article that talks about all of us. And I think a really great point is that, to really make sure that we're protecting these vulnerable populations and that we're not just understanding them from an outside lens, but from like a, we're here to learn from you, and vice versa, to make sure that we have indigenous researchers on those panels, making sure that when you go to those tribal councils, that they're just as informed as we are when we're making decisions.
Absolutely.
So I'm wondering if you've seen any pushes in academia or even in companies to do that as well.
Yeah. I mean, I'll give you my my sober view on this. I'm a little cynical on academia solving problems just because it has had a long time to solve problems and hasn't. And companies, I think, tend to move somewhat better in terms of social change. And I'll give you an example, not from my work, but actually Thomas Sowell's work on discrimination in housing and discrimination in education. And his view is that the administrators who were one day enforcing Jewish quotas in like 1967 turned around and became the affirmative action administrators in 1968. And I don't think he's wrong, right? Like, so on the other hand, if you look at companies and the way companies operated, it actually didn't make a lot of sense for companies to discriminate, right? Like, why would companies discriminate? Like, you happy to have a big customer base. And in fact, in many places, like the segregation that was imposed on the companies, not like the companies wanting separate counters, right? And shit like that. So there's this really complex interplay between what ends up happening and how academia does things and then how business does things. So my sense is that I'm getting so much interest from pharma in diversifying clinical trials in a real way. Like, how do we actually get this done? How do we achieve this? Whereas academia ends up like being just, who do we blame for this? And how do we, so I'm optimistic. I'm actually optimistic that this will happen because the science is so strong. And the right way to do things is not to steamroll people, but to actually say like, hey, we need to enroll folks. We got to get them on board. Like it's 2021. Come on, like, so I think this will change in the same way, like telemedicine just it's dramatically changing care delivery, maybe starting at the top, maybe it's not disrupting as quickly, but like pre-COVID, one of the things I was working on is telemedicine in the rural South and in the rural West. Like, I have to say telemedicine in New York is not as interesting a problem as telemedicine in Montana, right? Like drug delivery in New York is not as interesting a problem to me as drug delivery in Montana, So just because of the distances and it's an extraordinarily complex problem, right? When the closest town has one shop and it's a Walmart and a McDonald's, right? That is you want to talk about food, that's a desert within a desert, kind of stuff. And those are absolutely huge, massive knowledge inequality barriers that need to get totally, taken down. And they happen to, I think, be often overlapping in places. Just they're in the poorest counties, and they're in the counties that are also absolutely overrepresented with where you need to deal with both the knowledge inequality and the fact that you have to, and it's the right thing to engage the local tribal groups, right? So I don't know if that answered your question, Oliver, but I do think it'll move faster in industry. I think academia will try to do stuff, but I think it'll I'm not optimistic. Hopefully I'm wrong, but I'm not optimistic because it's a question of resources, and who's got resources? Industry has resources.
Okay, thanks.
Great. Well, thank you so much. I think we're just about out of time, unless there's, one other.
I'm happy, Pooja. I'll take your answer. I'll take your question and hopefully give you an answer.
Okay, great. Thank you so much. It's actually really related to Oliver's question, the answer you just gave. So hopefully I can just sort of tack it on there. But I'm a first-year PhD student. So nice to meet you. Thank you again for this talk. I was curious, sort of building off of what this knowledge and the knowledge and equity conversation you were talking about with regards to Oliver's question. If you've seen any best practices within academia or within industry, I'm thinking a lot for how we can use this data and give this back to participants and build those knowledge, build the knowledge within communities, and sort of start to close those gaps. I was curious how you started to see that change, and if you have any thoughts about how we can be doing that better. Specifically, I'm thinking about with regards to all of us and how we're leveraging that data, and also coming from this history of science as science specifically with marginalized groups as this very problematic space.
Excellent question. I think all of us is our national cohort. We should support it. Josh Denny is now the CEO of all of us. So the guy who I just said is doing the most interesting science work is also now the CEO of that state-owned company. It's a state-owned company. So I think we should really be, figuring out how we contribute to the creation of that cohort in a way that we're all proud of, right? Like I'm all for criticizing, and I criticized the 1,000 Genomes Project in order for it to be rethought, and I think there's an opportunity to do that with all of us as well. Hopefully that answered the question. Happy to answer more. Thank you for the opportunity. I really appreciate it. Sorry I couldn't be there in person.
Thank you so much. This was fabulous. We appreciate your time. Thank you.